Hi friends, I hope all of you are doing well. Today we are going to discuss about the SAR of the morphine and its analogues. We might be know the morphine, it will be comes under in the narcotic analgesic like morphine and various morphine derivatives like codeine, acetyl diacetyl morphine like heroin. All this will be comes under in the narcotic analgesics. Then how the SAR for this one, because this is very important topic for examination point of view. They are asking many times, and many of the students, you might be know, they are afraid to write the structure of the morphine. That is not the difficult part, but the thing is that it's happening like that. Why, we don't know actually. Maybe that is lack of practice or lack of concentration. Now directly we will go to our point. Then especially the morphine and its analogues. Then when it comes to its SAR, that is the relationship between the structure and activity, how the narcotic analgesic activity, it will be going to be changed based upon its structure, that we are going to be discuss. Now we'll draw the structure. It is having the phenanthrene derivatives in the basic structure. It's not the difficult part to write the structure. See, observe carefully how I am writing structure. This is the important aspect, and it's not the difficult to write also. Then see, in this case what exactly it is going to be happen, especially in the morphine and its derivatives. Any structure of the morphine you might be take. Here it will be having the ether linkage. Now all the narcotic analgesics. When comes to the morphine, it is having the OH group, here one OH group and here one OH group. Then it is completely aromatic. And it is having the side chain here. That will be attached to the nitrogen, that is the tertiary nitrogen. Here it is having the double bond. Now how exactly it is going to be change the activity when the change of the structure will be takes place. Here generally nothing is there, means hydrogen is there. See, when it comes to numbering of this one, according to the chemical abstract, we are starting the numbering from here. 1, then this is 2, this is 3, this is 4, this is 5, this is 6, this is 7, this is 8, 9 we are giving here, we are skipping this one. 9 is the 10, is the 11, 12, here the 13, here the 14, here the 15, here the 16. This is numbering, how we have to be give, that is based upon the chemical abstract. Okay, now see, in this case how the SAR it is going to be changed, the activity it is going to be changed based on this structure. What we are observing here in this case, you might be observing third position OH group. I will take, I mentioned here third position. This third position OH group, if we replace, okay, if we replace, you might be think with methoxy group or acetyl group, methoxy group or acetyl group or any other. Remember this, any other. Especially at the third position, what happening means it decreases the activity. Remember, decreases the activity. Which position I told? Especially third position. We are not discussing every position, means which is not participating in the binding with the especially D2 receptors, which will be shows the action on the central nervous system, dopamine receptors. Then this position, either you can be replace this OH group with methoxy or acetyl or any other, the activity is going to be reduced. That's why you might be observing this methoxy group, it is present in the codeine. Present in the codeine. When it comes to the codeine structure, only the difference is here. If we want, we can write one more time here also. I will write the structure. See, just observe how I am drawing this structure. Now I am taking from here, not from here. Now again we are coming in this sequence. Here the ether linkage as usual. Here the difference is instead of hydroxy we are having the methoxy. Here it will be same. This is all about, we are discussing about the coding. Okay. The aromatic, here the double bond in between the 7th and 8th position. This is called as the coding. What is the difference? Only the methoxy group and hydroxy group. See, if you know the structure, drawing this structure, only the morphine, you can draw the near about 8 to 9 structures which are belongs to the specially narcotic analgesics. We might be know this narcotic analgesic, non-narcotic analgesic, two things we will be have. In case of narcotic, these are the addiction, it causes the addiction. Because it will be belongs to the narcotics, that's why it causes the addiction. And because they are working on the central nervous system especially, not only that, we are using in case of general anesthetics also working on the central nervous system. Okay, you might be know all these things. Non-narcotics, they are not causes any addiction. You might be have the aspirin, you might be have the paracetamol, diclofenac sodium, all they are comes under in the non-narcotic. But these are the addiction, it causes the addiction because they are belongs to the class of narcotics. Now see, particularly in this case, what I told, this third position methoxy group. Now we can go for the ether linkage, where we are observing here the ether linkage, this one. Why I am calling ether linkage? We might be know ether functional group ROR. This is very important here, ether linkage. If we remove this ether linkage, remember this. If we remove this ether linkage, okay, then what happens means removal of the ether linkage increases the activity. Why means here especially removal of this ether linkage, what produces means morphine and derivatives. Remember this, morphine and derivatives. This morphine and derivatives especially 10 to 100 times more active. How many times? 10 to 100 times when compared to the morphine. This is more active because removal of this ether linkage. Removal of the ether linkage means there is no ether linkage. There is no ether linkage. Then I will write here. No ether linkage means this one. If you remove this ether linkage, it produces the morphine and derivatives. They are the 10 to 100 times more active when compared to the natural morphine. Okay, because these are the synthetics. Morphines are the synthetic, semi-synthetic. They will be comes under in the class of synthetic as well as the semi-synthetic. Now, this point is over. Ether linkage. Will go for the sixth position. It is a little bit different. Why means you might be observing the sixth position. Either you can be do the acetylation. Remember what? Acetylation you can do at the sixth position, and we can do the oxidation. Okay, either you can do acetylation or you can do the oxidation or you can do any other process. Especially here, any changes will be occur here. Okay, that may be oxidation, either it may be acetylation, either it may be any methylation or anything you can do here. What happens? This one means it increases the activity. See the difference between the third as well as the sixth position. Third position, if you do the oxidation or means acetylation, methylation or anything, what happening? The activity it is going to be reduces. But in case of sixth position, here if you convert this one by the oxidation into the ketonic group, the double bond O, hydrogen will be goes out. That if you will be do that process, then you might be know the activity increases. Acetylation means this hydrogen will be replaced with the acetyl group, that also activity it is going to be increases. Okay, if it will be replaced with the acetyl group or oxidation or anything, here any changes, it is going to be increases the activity. That is the difference between the especially third and sixth position. Now comes to the this in between 7th and 8th position. What is there? Double bond is there. This double bond is necessary. Remember this. Necessary for optimum analgesic activity. Remember this point. Okay. Which position? 7 and 8. Because that's why we are here doing the reduction, that especially introduction of the double bond, it is helpful for the increasing the activity, means it is necessary to produce the optimum analgesic activity. Here 14th position you might be observing, this one you can replace with the OH group. If you replace with this hydroxy group, only hydroxy group, not any other group, increases narcotic and analgesic activity. Remember this, narcotic and analgesic activity will be increases, especially at the 14th position, hydrogen will be replaced with the hydroxy group. See how much it is important. Now you can see the amazing thing, especially at here the nitrogen at this position. If you replace this methyl group here, if we replace this methyl group especially with phenylethyl group, or this N position, this methyl group itself. Okay, if you will be think here the R is there, if this R it is going to be replaced with this ethylamine or it will be replaced with the methyl, then here what will be happening means it produces the agonist activity. Agonist activity means narcotic analgesic activity, means increases the especially the effect of the D2 receptors here. Okay, that's why the analgesic and narcotic activity it will be increases. But at the same position you can see, at the same R position I will write here, if it will be have the any allyl group, listen carefully, if it will be have any allyl group or if it will be have any cyclopropyl group, this cyclopropyl group, this produces the antagonist activity, not the agonist. Antagonist activity. Antagonist activity means decreases the narcotic analgesic activity, means it will be counteract the narcotic analgesic. Here agonist, either methyl or phenylethyl agonist activity, means narcotic analgesic activity increasing. But here if you take the any allyl group or cyclopropyl group, it produces completely antagonist effect. This one we are using to reduce the effect of the narcotic analgesic when it will be have the allyl group or when it will be having the cyclopropyl group at this R position. That's why this R position is playing the vital role, very vital role. If you want to produce the agonist or antagonist, that depend on this position. Okay, I hope you might be understood this one. This is the SAR of the morphine and its analogues. Remember this, if here you can be replace this both the hydrogens especially with the acetyl group, that's called as the OCO CH3 and OCO CH3. Remember this. In this case, what happened? Here the ether linkage will be there. This is called as the diacetyl morphine. Diacetyl morphine, and it is also called as another name is the heroin. This is we are preparing from this morphine. How we are preparing means by the process of the acetylation. We required here acetic anhydride. See, the replacement of both the hydrogens from the morphine here. Here hydrogen is there, here hydrogen is there in morphine. The replacement of both this hydrogen with the acetyl group. But the activity what happened? Decreases. What activity? Decreases the narcotic analgesic activity. Remember this. Like this, we can be doing any modification on the morphine specially, which we are obtaining naturally. Any modification, some positions, here it will be decreases, here it will be produces morphine and removal of the ether linkage as I shown. If you remove this oxygen, morphine and products produced, they are the more active, 10 to 100 folds more activity when compared to this morphine. Here also it will be increases the activity, either acetylation, oxidation or any other process, methylation, all they are going to be increase the activity. Double bond, it is necessary for the activity. Hydrogen you can replace with the hydroxy, it will be increases the narcotic analgesic activity. Here our position is very important. Okay, if it will be the phenyl ethyl agonist, methyl agonist, if it will be have the allyl and cyclopropyl, it produces the antagonist activity. This is about the SAR of the morphine and its analogues. Please try to draw the structure, then writing part it will become very easy. Okay, thank you very much. Take care. Please subscribe the channel. With subscribing, nothing will be going to happen, nothing we are going to be lose. I hope you will be cooperate. Thank you very much.